Morning students, today we will study the bacterial cell wall. Underlying the capsule and external to the cytoplasmic membrane in the cell wall which provide rigidity and shape to the bacterial cell. Without their cell wall, all bacteria assume a spherical shape. The most important function of the cell wall is to protect the cell content from the external stress and from lysis resulting from osmotic pressure. The cell wall is made up of peptidoglycan, also known as murein and mucopeptide, made up of sugar and amino acid. A single layer of peptidoglycan is a network of adjacent sugar and acetyl glucosamine bound together through the amino acid side chain. Thus, peptidoglycan is a highly cross-linked molecule which give an amazing strength to the cell wall. The bacteria fall into two major natural groups according to their reaction to a strain devised by Christian Graham in 1884. The two groups of bacteria, that is the gram-positive and gram-negative bacteria, differ mainly in their cell wall. Before considering its structure and function, it is important to know about the differential staining method, the Gram's stain. Bacterial staining. Gram stain bacteria can be divided into two large groups on the basis of differential staining technique called the Gram stain, developed by a Danish physician Christian Gram in 1884. One large group is called the gram positive and the other is gram negative. These two groups that are morphologically indistinguishable yet it differ in their cell wall structure and metabolic character. In this process, a heat fixed smear of bacteria is stained with crystal violet dye for 30 seconds. After rinsing off, a dilute solution of iodine is added. All the bacteria are stained deeply blue. The smear is then treated with 95% alcohol. Some bacteria are completely discolored by alcohol, while others retain the crystal violet dye. The bacteria are finally treated with some counter stains such as safranin. Two types of results are noted. Those species of bacteria that are discolored by alcohol appear red in color. These are the gram negative, and those stain retain the purple primary stain are called the gram positive. The cell wall of the gram-positive bacteria. In gram-positive bacteria, the cell wall is composed of several layers of peptidoglycan forming a thick, rigid structure. The cell wall has a high content of peptidoglycan, that is 20 to 80% of the dry weight of the cell wall. In addition to peptidoglycan, the cell wall of gram-positive bacteria contain teichoic acid, large molecule composed of repeated unit of sugar and phosphate. These acids contribute to the cell wall strength and prevent possible cell lysis. The gram-negative bacteria, the cell wall of the gram-negative bacteria is more complex than the gram-positive bacteria. In gram-negative bacteria, cell wall beyond the cytoplasmic membrane lie the periplasmic space bounded on the outer side by a thin layer of peptidoglycan exterior to which they possess a second membrane, outer membrane. The periplasmic space is an aqueous space having the consistency of a gel with a loose network of peptidoglycan running through it. The peptidoglycan of gram-negative bacteria has the same functional structure as found in the gram-positive cell, but gram-negative cell generally have only a thin layer of peptidoglycan. The outer surface of the peptidoglycan is covered by a series of layers which provides a protective coating around the cell. The first of these layers is lipoprotein, which provide an anchor for the outer membrane. The outer membrane is a lipid protein bilayer similar to the cell membrane. This layer protects the gram-negative bacteria from the effect of antipeptidoglycan chemicals such as penicillin and lysozymes that could damage the cell. As a result, infection from gram-negative bacteria are often more difficult to treat. The surface of the outer membrane contains molecules of lipopolysaccharide which has a toxic property. Thank you.